Hello everyone, and welcome to our online lecture series for Bio 1. Today we'll wrap up our discussion of the nervous system by talking about our sensory systems. Now, of course, our sensory systems rely on the signal sending that we saw in our discussion of neural networks last time. That signal sending relying on the difference in ion concentration between the outside and the inside of cells. So as a quick review of that process, we know that sodium ions are at high concentration on the outside and potassium ions are in high concentration on the inside. In order to send a signal from this resting potential, we'll need three different players: the sodium ion channel, the sodium-potassium pump, and the potassium ion channel. So two proteins that are specific to each ion, and then one that does the pumping afterwards to reestablish this resting potential. Now, in the rising phase, the beginning of the signal sending, we see that sodium ions are going to be rushing into the cell. Keep in mind that these sodium ions have a positive charge. When that positive ion pressure starts coming in, they fall down their concentration gradient because diffusion will want them to even out. And this causes a change in the in the overall charge on the inside of the cell. Now, once the concentration of sodium ions reaches a certain level and changes the overall charge on the inside of the cell, we see a change in the conformation of our two channels. The sodium channel will close to prevent any further sodium from coming in, and instead the potassium channel will open. And in opening, diffusion now pushes potassium on the other side of the cell. This causes, again, a flux in the voltage across the membrane and a change in the ion concentrations of each side. In this depolarization, we then reach a hyperpolarization or subsequent repolarization, wherein the ions are fairly mixed on both sides, but this is very, very, very far away from our resting potential as seen above. Instead, once the potassium ions have gone back out and the sodium ions have gone back in, they now remain closed to prevent diffusion from happening, and the transporter, the ATP-driven transporter, can begin to push sodium ions out 3 at a time and potassium ions in 2 at a time, getting us back to that resting potential state. So all of our signal sending today is going to rely on the same kind of ion transport that we see in the basic neuron. And just as a last illustration of this process, we know that this is occurring down the body of the axon, where certain regions begin as being depolarized at first, then that signal will transport adjacent regions that were at resting potential, with repolarization happening behind. So you really do get this stepwise pattern of the action potential traveling down the neuron. And in this way, you can send signals very quickly, one after the other, but notably not simultaneously. Because from last class, you will remember the things that stop signals from interfering with one another. If you don't, please make sure you go back and pay attention to that, as well as the graph of voltage, because you may be asked one way or the other to explain either the movement of ions across these membranes in an illustration form as seen here, or in the form of a graph. Now, our sensory organs, our sensing systems, come in 5 different forms. And all of these forms rely on turning some sort of mechanical or chemical signal into an electrical signal inside of your cells. And all of this occurs via signal transduction of different types. Now, of course, you will be familiar with your 5 different senses: the sense of olfaction, the sense of gustation, the sense of equilibrium, and then the 2 that are fairly straightforward: the sense of hearing and the sense of vision. Equilibrium is your sense of balance. Gustation is your sense of taste. And olfaction is your sense of smell. And keep in mind that all 5 of these operate on this principle of turning a mechanical or a chemical stimulus into some sort of electrical stimulus within your neurons. The signal transduction types that we see within these 5 different senses vary. They either are a team effort, meaning that there is some sort of neuron plus receptor, because you do need some sort of specific way of detecting things like the elements that make you smell or the elements that make you taste. And these generally fire a little bit slower because there's 2 cells involved, and they're always responding to specific stimuli. And they're specific to one type of stimulus, because that receptor, your balance receptor, will not be able to taste foods. They do not cross-reference one another. The second type of signal transduction that we see is a free nerve ending. Some sort of way of immediately transferring a signal, and this is most commonly seen in things like pain. You'll notice that pain is a very quick sense for you to detect because you obviously want to limit how much pain you expose your body to. Broadly speaking, there are 3 different stages in which you sense things. Now, in order to sense, you obviously will begin, stages towards sensory perception. You first have a sensation. The sensation will rely on some sort of stimulus in the perceptive field. And all this means is that, of course, in order to detect touch, something has to poke you in a place where you have the distance to detect. So if a poke is performed far enough away from the body that it does not actually touch the body, you will not feel that poke. You might be able to see it, but you won't ever feel it, right? It does not travel through the air. In the same way, someone cooking 2 houses down, even though that smell is created, because it is not within your receptive field, you will not be able to sense it. Step 2 is some sort of transduction. This, of course, being sending that physical signal or chemical signal into an electrical signal. And of course, you have our usually specialized mechanoreceptors to one type of input, but your brain can eventually combine these individual receptors to some higher level function, such as with taste, because you know that foods have different mouth feel as well as flavors. So our brain is able to take these different transduction signals and combine them all into one perception, which is step 3. And in perception, the brain, specifically the thalamus, processes your signals. So these are the things that are required in order to have you be able to sense things in the first place. But there's one other principle called the all or none principle that will apply to our ability to detect things. The general idea behind the all or none principle is illustrated by these graphs. You have first some sort of strength of stimulus, so how strong the thing is that you are detecting, how intense is the poke, how strong is the smell. And we see that weak smells or weak pokes will elicit no response. Once you reach some sort of threshold that can incite the action potential, you get a full response. Touch is detected, smell is detected. So in this way, we have kind of the inability to tell small variations between the strength of the stimulus. This is happening at the single neuron level. A single neuron can't tell how strong something is. It can tell you whether it's there or not. Your brain will then potentially perceive things later on by seeing how many neurons are detecting it, so that gives you the idea of strength. But the all or none principle certainly applies to single nerve cells. And this relies on the fact that the action potential, that ability to move sodium ions in or out, will either be triggered or it won't be. It does not change how much ion is moving inside or out. It either happens or it doesn't. So biochemically, on a cellular level, you understand why we see the all or none principle coming into effect. It's action principle independent of strength of stimulus. And there are 4 components to being able to detect the strength of the stimulus. The first is the type. Do you actually match the signal with the receptor that's present? The location in your field, meaning is it close enough to your receptor to be able to be detected? Assuming your receptor is correct. The duration of the signal, is it persistent enough to actually incite the action potential? And number 4, the intensity, which relates directly to the signal strength itself. So taking these broad ideas behind the stages of sensory signal and the fact that they will either be triggered or they are not, we can talk about 3 different senses specifically today. Now, in a kind of arbitrary fashion, I will be talking about taste, touch, and vision in that order. So for the purposes of the exam, I don't need to pay as much attention to the sense of smell, nor hearing. But they are mentioned in your book, so please feel free to read about them. Let's begin with taste, which is my favorite. Taste is a sense that relies both on mechanical stimulus and chemical stimulus. And we'll talk about how exactly the receptors process this. Taste has 5 different types of receptors, and therefore we have 5 different flavors that we can perceive. Those are sweet, sour, bitter, love bitter, salty, and umami, or the earthy flavor, savory flavors. The best illustration of an umami flavor occurs when you put salt on a tomato. When you put salt on a tomato, you create a very particular type of salt called monosodium glutamate. Now, you may be familiar with monosodium glutamate, or MSG, as it is a common additive in Chinese food and previously was blamed for some Chinese restaurant headache that occurs afterwards, but there is no evidence to support that the MSG salt is actually the culprit here. And maybe that Chinese food is quite tasty and we happen to eat just too much of it. But monosodium glutamate does not seem to have any adverse health effects in blind controlled studies. Plus, you get it when you add salt to tomatoes. Of course, MSG is added frequently to different meat dishes because it enhances that umami or savory flavor. Now, each of these different flavors are detected on our taste buds. Which have these different forms of papillae or hairs, the fungiform, the filiform, the foliate, or the circumvallate papillae. So different types of hairs that allow for different types of detection. But ultimately they wire into something where you have a gustatory cell, meaning a taste bud cell, and the hairs and the pores on the top that either allow for mechanical sensation or chemical sensation of different types of flavor. The smells that are associated with food actually come from specific chemicals in the olfactory neurons, those being those bipolar neurons that are olfactory. And that will have been discussed last time. In contrast to those, our taste buds rely on different chemical signals. Our sweet flavors will rely on a G protein coupled receptor, so a signal that is actually able to transverse across the membrane. Or G proteins are often referred to as molecular switches. They use GTP and GDP to turn on signals or turn off signals inside or outside the cell. Our sour receptors rely on acids that have hydrogen ions. Bitter and salty are also G protein coupled. Bitter is G protein coupled. Salty is actually derived from that sodium itself. It provides the sodium that will venture into the channel, which is quite a clever way to do things. And umami also relies on G protein coupled reception. So you have different signals that begin this transduction, whether they are G protein coupled, like in the sweet, bitter, and umami, or acidic hydrogen ions in sour flavors, or sodium ions in salty flavors. Ultimately, all of these different receptors are processed by our taste buds on our tongue, or received by our taste buds on our tongue. The signal is transduced into the brain, where it is perceived as a combination of both mouth feel, or the texture of your food, and the flavor from these 5 different receptors. So you have things that are both crunchy and salty, combinations of both mechanical and chemical reception. So next up, we will talk about our sense of somatosensation. Oops. That being our ability to respond to stimuli such as temperature, pain, and pressure or pushing. Within our broad category of somatosensation, we have a subset of it called vestibular sensation. This corresponds directly to spatial orientation. And proprioception. Proprioception. This being our ability to tell our position of our limbs. Some of us are better than others at this, and that's why some folks may consider themselves clumsy, like myself. Proprioception has the movement of those limbs called kinesthesia, with this being an E here, apologies for my handwriting. Kinesthesia, limb movement. And ultimately, this limb movement relies on a couple of different cellular features. The 2 features that control kinesthesia are first, spindle fibers in muscle, because your body can detect the movement of spindle in your muscle as a stretching of the muscle, so you physically feel your muscle stretch. And then there is a specific organ that detects contraction, and this is the Golgi tendon. Not to be confused with the Golgi body, because this one is actually detecting a movement pattern in kinesthesia. Now, these components of somatosensation have similar interplays with what we detect as pain. Pain is a sensation that is processed in the brain, and I will provide proof of this. One example of something that gives you pain that should not, in theory, give you pain is the idea of a hot pepper. Peppers are hot because of a very specific enzyme called capsaicin. Capsaicin tricks your receptors on your tongue to be flooded with calcium. Now, calcium gets flooded in when those mechanical receptors are detecting changes in heat. They're literally feeling as though you just pressed your tongue against a hot iron. But they're not. It's not a hot iron. You're not getting burned, but the sensation is exactly the same because the signaling molecule is the same here. It's the rush of calcium. And capsaicin is literally just triggering us to feel heat even though heat is not present. It's playing with that mechanical receptors. It also, at the same time, causes you to shoot a signal to your fight or fight or flight response. Because like if you were trying to put your tongue against the hot iron, your body would be very, very heightened in its alert level and try to make you run away from it. And so you will notice that eating hot foods not only makes you feel, or eating foods with capsaicin that are not hot but rather spicy, will make you feel as though you are pressing your tongue to something fiery, but also induce panic because you think you're about to get in a fight for your life. Hence why your eyes might bulge, your senses might stimulate, and you might go a little crazy to try to deal with that very, very hot ghost pepper. I absolutely adore hot flavors, and despite the fact that I grew up with no spicy foods, I do like to make the joke that in my family, potatoes may have been considered a spicy food if too much table pepper was added to it. Over the years within college and subsequent life afterwards, I have continued to cook hotter and hotter food with hotter peppers and have gotten used to it because that overstimulation with capsaicin has caused my receptors to be less sensitive to it. So you will perhaps be familiar with the idea that you can build up your ability to process pain in the form of heat, but that's actually because your receptors are getting more used to that level of excitation, not that they're getting stronger and more resilient, they're just getting to ignore it more. Now, your book gives a very, very good illustration of the 5 different types of mechanical receptor endings that have to do with different feelings of pain or touch. These are our Merkel's discs. Merkel discs, being at the surface, respond to light touch. Our Meissner's corpuscles that are also more towards the surface of our skin, the epidermis, and these also respond to touch, but also low-frequency vibration, so you can feel shaking. Ruffini endings and our 2 other receptors are embedded a little bit deeper in the skin. Ruffini endings detect stretch and deformation of joints as their shape would indicate. They detect our movement of limbs, as well as warmth. So you feel warmth in terms of distance from the skin. You feel it much closer to your skin than you will something like cold. Our Krauss end bulbs detect cold. They're also in the surface, and then, I should not say you detect hot and cold in similar fashions because they're about to the same depth level, with Krauss end bulbs being our cold receptors and Ruffini endings being our warm receptors. And then the ones that are the furthest, deepest within our skin are our Pacinian corpuscles. These detecting changes in pressure, so something that pokes you but then stops poking you, as well as high-frequency vibrations. Now, all these different receptors are what we use in order to form our senses of somatosensation, vestibular sensation, proprioception, and ultimately limb movement. So, with our discussions of touch and taste, we now can go on to, I think, probably our most unique and cool sense, our vision. And I'll preface this all by saying that vision operates on a very different signal sending pattern than our other senses. And hopefully that'll make sense to you by the end of our discussion today. So, vision occurs in our eyes. The eye is a very, very complex organ, even if it is very small. The outermost layer of the eye is the cornea, which in effect is an eye cover that refracts light. Oops. In addition to the cornea, you've got the pupil, which is actually the hole that allows light in. And the iris, that is essentially the diaphragm that controls the size of the pupil. The iris is also where pigment proteins are expressed, so the iris is where you get color. In between the cornea and the iris and pupil, you have the aqueous humor, or a liquid layer. And then you have the lens that allows for the focusing of light. Once you get to the inside of the eyeball, you have the vitreous humor, or the large sac of liquid that keeps the eye inflated, giving it its round shape. And you have the back of the eye, the retina, that is going to be receiving light and converting it to vision. And then the fovea. The fovea is a region in the back of the eye that's responsible for acute vision, or very focused vision. It has a high density of cones, meaning that it is highly perceptive to differences in color. So you'll notice that when you focus on something, you can distinguish different colors much better. But also, it has the ability to move, so that way you can focus on different things. So when your eye moves, it's not just the lens moving, as well as the cornea, pupil, and iris, but rather the whole thing is moving, so that way the fovea is staring directly at the thing that you want to focus on. The entire eye itself feeds into a single optic nerve. That optic nerve will travel to the brain to allow for those signals to be transformed from light stimuli to some sort of vision sensory cells. Now, within the retina, there's actually a lot of complicated features, but I'm not going to ask you to remember things about the ganglion, the amacrine, the bipolar or horizontal cells. And instead, we'll focus heavily on rods and cones for our next section. Now, rods are light receptors that focus on peripheral vision and night vision. They are focused on the outer edges of the retina, rather than being embedded in the back. And in this way, they're more detecting presence or absence of light. So when you think about your night vision, you know that it's not maybe the best in terms of differentiating colors, but you can tell presence or absence of light. And that's because it's the rods that are doing this. They're not made to detect differences in color, they detect presence or absence of light. So in the dark, everything looks a shade of black to white. Cones are very different and quite fascinating, both from a physiological and a genetic perspective. Cones are actually very weakly sensitive. They're focused on the center of the retina, and they detect bright lights because you want that, or rather you want them detecting bright lights because they are weakly sensitive. They are going to be able to detect these major fluctuations in the presence of light, but also the presence of different colors. And you actually have what is called trichromatic vision, because you have 3 different types of cones, and each of these cones specializes on a different wavelength of light. You have S cones, M cones, and L cones. They are named very cleverly because they either focus on short wavelengths of light, like blue light, medium wavelengths of light, like green light, or long wavelengths of light, like red light. And in humans, we see this prevalence of trichromatic or full color vision. Contrast this with monochromatic, dichromatic, our trichromatic, the occasional tetrachromatic vision, and even the pentachromatic vision that's seen in some other species. So organisms with monochromatic vision are really only able to perceive low light. They're adapted to low light environments. They have one cone and can only perceive something like 200 colors, and they're therefore specialized on these low light environments. So when you think about these, think of marine mammals. Organisms with dichromatic vision have 2 types of cones and can perceive something like 40,000 different colors. Now, most terrestrial mammals that are not primates have dichromatic vision, as well as colorblind primates, including that of my siblings. They both suffer from some different color sensitivity, so they're not truly dichromatic, but they are red-green colorblind. And there are, of course, humans who are truly colorblind and other primates that are truly colorblind. But whenever I think of my dichromatic visioned friends, I always think of the dog currently sleeping and snoring on my feet. We've heard dichromatic vision compared to night vision, again because you're seeing presence absence and very little differentiation in color. Once we get to trichromatic vision, that of course is again the 3 cones that we see, the L, M, and S cones, and we see up to 10 million different colors. Now this type of trichromatic vision is common amongst the primates, the marsupials, and some insects. Honeybees, for example, have trichromatic vision. That's why we oftentimes see pictures of what honeybees see flowers like, seeing the landing pads there. But it is possible to have the wavelengths of those light skew more towards ultraviolet. And that's what we see within the bees. Even though they are trichromatic, rather than having the middle cone being something in the green, they shift towards even shorter wavelengths of light to be able to detect colors that we do not see, and we detect colors that they don't. Now, tetrachromatic vision means that you have 4 different cones, and there are humans, rarely, that have tetrachromacy. However, a lot of reptiles, amphibians, birds, and insects see with 4 different types of cone cells. With 4 different types of cone cells, you're able to detect 100 million different colors. And I'll take a moment here to explain how this is possible in humans. The gene for cones, particularly the M and the L cones, are present on the X chromosome. This is one of the reasons why we see color blindness being more prevalent in males. Males are XY, females are XX. Therefore, if a male has a broken version of an L cone on their X chromosome, and there are tons of broken versions of genes on all of our different chromosomes, they don't have a second copy to give them that color vision. In contrast, females are oftentimes thought to perceive more colors not because of who they are in terms of personality, but because they have two X chromosomes and are more likely to have a working copy of the M and L, particularly the L type cone. Now, it is possible for some females to have different alleles of the L type cone. And if you have two different alleles of the L type cone, you not only have S, M, and L, but you have L1 and L2 type cones. And in females that have these four different color receptors, these four different cone cell types, they actually have tetrachromacy and do perceive those hundreds of millions of colors. But this is actually very rare within females. So, yes, it is possible for some females to have an advanced form of color detection. But keep in mind that tetrachromacy is actually common in reptiles, amphibians, birds, and insects. Pentachromacy, with 5 cone types, actually allows you to perceive 10 billion colors and is not as widespread, but we do know that there are some insects, such as butterflies, that have 5 different types of cones. Gives you new perspective as to how much variation in color that butterfly must perceive in its own beautiful wings. And then some birds have pentachromacy, including pigeons. The bird that we oftentimes think of as the rat of the sky maybe doesn't need to be so pretty or glorious in itself because it sees the world in all of its beautiful colors. Kind of a poetic way to think about pigeons, I think. Now, this variation in presence or absence of different types of cones and different color perception obviously has to translate into some way for sending that signal. And the blueprint for rods and cones is actually fairly similar. This is an example of a rod cell that has this receptor at the top, and this receptor is modeled here. If you wanted to see what a cone cell looks like, you replace that top rod part with photopigments. Those photopigments being those M, L, and S that focus on different wavelengths of light. In these rod cells, we need to talk about the process of signal transduction. Because you'll notice one very unique thing. In its native state, when the rod cell is not actually detecting, it is sending a signal of glutamate. The default state in darkness is for the rod cell to be sending a signal to its bipolar cell. This means that darkness is the presence of a signal. So what happens when light actually hits the rod cell? Well, the rod is able to detect it with a very specific protein complex called rhodopsin. Rhodopsin itself is made up of 2 different proteins, opsin, that spans the membrane, and retinol. So opsin is the thing that goes across the membrane. Oops, not 3 S's. And retinol changes shape. So what happens here is that light will come in, hit the rhodopsin. When the rhodopsin is hit, it changes transducin, which is a G protein, that will then go and impact phosphodiesterase. So you have phosphodiesterase and transducin interacting with one another. It hits the phosphodiesterase with a GTP molecule, so not ATP, but guanine triphosphate, and that GTP will then cut off the cyclic GMP that's attached to a protein channel, to a sodium channel protein that causes that sodium to constantly be letting in. And if you cleave off that cyclic GMP signal, you close the channel. When the channel's closed, you cause hyperpolarization. In that hyperpolarization, you're able to detect light and detect quantities of light. So that's a lot to unpack. Let's try to go over it again and understand the biological significance of how this works. Your rod cells are constantly sending a signal saying dark, 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 dark. When light hits them, you get changes inside of the cell to reduce the amount of sodium that's being sent in by closing those channels. So rather than saying dark, 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 it might be going dark light, 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 causing a hyperpolarization in your bipolar cell that then allows you to detect the presence of light. And in this way, by screaming dark very loud and then transitioning to a whisper of light, 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 you then get this variation in intensity. And you notice this difference, right? If the lights are on very, very bright, you're able to detect that. You're not just detecting the presence or absence of light. And this is entirely due to this mechanical method of always be sending a signal and reduce the intensity of that signal in order to allow for variation in the intensity of light. Now, on the inside, this is all driven by that transducin and phosphodiesterase and cyclic GMP that interact to change what's going on with our sodium channel. And in this way, by vision always screaming dark and getting quieter and quieter as light enters the scene, we are able to detect variations in light intensity as well as variations in color. Intensities coming from our rods, colors coming from our cones. Now, this means that ultimately vision is actually the perception in the brain of less of your neurotransmitter glutamate. And the big advantage of this is that you get a much stronger, noiseless signal that allows you to detect even single photons of light. You all know this. If you're in a dark room for a while, and then all of a sudden the faintest bit of light enters, you're able to detect it, which is pretty cool. So we'll end with this summary of the comparison of rods and cones. Rods are used for scotopic vision or under low light conditions, whereas cones are under high light conditions, meaning that they're weakly sensitive. They're not very sensitive compared to very light sensitive. If you lose your rods, it can cause you to be blind in the night, Whereas loss of cone function, it leads to legal blindness, inability to detect colors. Rods are not present in that focus feature, the fovea, whereas cones are highly concentrated in the fovea. And then all the other different features of color vision versus achromatic vision, one type of photosensitive pigment or 3 types of photosensitive pigment, and more in other organisms. Their counts differ. You have fewer cones and many more rods across the retina. But ultimately, hopefully, all of those are intact. So, hopefully, this explanation of very complicated vision, vision being the absence of a signal or less of your neurotransmitter, really stands in contrast to signals being sent by any of your touch receptors, touch or vibration or warm or heat receptors, or your taste receptors, which are quite my favorite, are 5 different tastes. And that all of this operates on some sort of all or none principle. Please keep in mind that everything you've learned in terms of neurons, with signal transduction and the potentials across the membrane, are absolutely critical in understanding how your nervous system does so many different things and processes so many different signals, and will leave out anything about the processing inside of the brain. We're just focusing on how our mechanical receptors allow us to have our different senses, our 5 different senses.